to the which is talking about the use of non-RCT studies in regulatory decision making. I'm David Wright from AstraZeneca, and is Iftika from EMA?
That's right.
Yes, who is my co-chair.
So welcome from me as well. Just a bit of housekeeping, especially for the people joining us online. Just to remind you to write your questions to the Q&A functionality. Please do not use the chat because we don't monitor that. And with that, I would like to leave the floor to David for the introduction of our first speaker.
Thank you. So it's a great pleasure to welcome Kit Rose to the stage, who's Professor of Biostats at the Rabun University Medical Center and also Chair of the EMA's Methodology Working Party. So over to you, Kit.
Thanks very much, and also for here have the opportunity to present on single-arm trials. I'm guessing, I'm actually anticipating that I will be sharing a reflection paper that quite a few of you actually have looked at and maybe critically looked at and have or will be sharing comments to fairly soon. So I tend to be fairly efficient and brief in my presentation. Need this one. Does this actually work to move the slides? Yeah, it's a new algorithm for me. So the motivation for having a reflection paper on single-arm trials comes from the fact that if you look at the marketing authorizations, dossiers over a longer period of time, actually a substantial and relevant number of applications base the primary pivotal evidence on single-arm trials. I think this is particularly well known and seen in oncology, but if you look closely, it's across more than oncologies. It's across different therapeutic areas. And we see it as a recurring challenge to actually properly assess single-arm trials and the level of evidence that's actually there. There is no guidance which essentially helps us. And of course, also we would like to be consistent in the way we approach such development programs. And we see a need to indeed communicate the challenges. And here I'm actually hoping to a large extent being praying to the convicted, so that you think we possibly share the same view on the challenge with sets, but we also think that this reflection paper can be used to actually improve the design and context, and also some of the discussions we had earlier, the why question. So why actually go for a single-arm trial, and why do you actually think it provides solid evidence? And it's a reflection paper because we really want to engage in the discussion. We're not there yet. It's not a reflection paper. Yeah, of course you read it like that, but it is of course not guidance. It's a reflection on a brief explanation on how we developed it. It's on the work plan of the CHMP, and not only of the CHMP, but also associated committees. So particularly the one on advanced therapies, where it's also quite common to see single-arm trials, and the oncology working party, because we are quite aware that that's an area where it's seen relatively more than in some of the other areas. It's not a guideline or a reflection paper written only by statisticians. So the group that worked on the reflection paper had representatives there from the CHMP, from CAT, from a clinical perspective. And we kind of worked with the core group with 2-3 clinicians, 2-3 statisticians, and a wider group. And that was particularly wider, both for the statistical sense. So there we actually used the Biostats Working Party, as well as on the clinical side, more members of the CHMP and the CAT to also critically review, I've not counted, but some versions of the reflection paper. And just as a tongue-in-cheek, the original title of the reflection paper was The Importance of Randomization. Didn't make it as a title. A crucial consideration, at least on how we wrote the reflection paper, but probably also how it needs to be read. It's a single-armed trial, and you write a protocol, and the single-arm trial is there. And that's what you submit to an ethical committee or to a seat for a CTA, and then you start a study. So the way we've looked at it how do we look at a single arm trial as a well-defined, on its own standing experiment? Because that's the way how it's proposed. If you can, of course, come up with other designs. I write a protocol in which I concurrently also have non-randomized control data there, but that's a different design. So we very much took the position, to what extent are we able to design, interpret a single arm trial as an experiment on its own. Usually if I talk about experiments, I make a copy of this book. It's not a new book, it's by Sir David Cox, probably the 60s of last century. And I can actually promote it to anybody ever being involved in experiments to actually read it. And it's not very technical, but probably you only understand it if you also have some experience there. But it's experimental thinking. For single arm trials, there is, let's say, two angles there: methodological conditions to establish a treatment effect, that was actually treatment effect, and that we can causally attribute it to the treatment. So that's question number one that we had in our minds. The second one, we're not there yet, is actually considerations, and their limitations become even more impactful to estimate the treatment effect in the population of interest. And those two, of course, are not exactly the same thing. And I think those two angles are there in the reflection paper. So in scope, methodological consideration across all therapeutic areas. It might happen that there are specific therapeutic additional reflections or guidance developed over time. So it needs to be overarching. Pivotal evidence and efficacy, and across design and up until assessment. So nothing therapeutic area specific. I think we mentioned safety briefly, but do not deal with it extensively. But I think we can all imagine that there's also some challenge there with an uncontrolled design. And also not considerations on feasibility of RCTs. But pretty much we were, of course, triggered. So can you actually indicate under which conditions that this, you kind of accept that there's no other way than a single arm trial, which usually is called, yeah, well, but an RCT is not feasible. We did not go there, and I think in my mind, there's a very brief thought. Feasibility is actually quite independent from whether or not a single arm trial will give reliable evidence. So for the methodological considerations, I actually consider feasibility not a very helpful thought, because that doesn't determine whether the single arm trial you subsequently propose provides reliable evidence. That's a very independent consideration. Important, but has nothing to do with evidence generated. So here I want to be really brief, because I think there it's the praying to the convicted. Lots of obvious deficiencies if you do a single arm trial. No concurrent control, no randomization, no blinding of anybody. The enrollment is not unconditional, so it's not also there. The randomization is lacking. It's enrolling for this particular treatment and not, let's say, unconditional into a trial that investigates the effects. So the randomization lacks in terms of using randomization as also a means to support causality. I will come back to causality later on. Of course, there are bias. Reliance on external information and assumptions is essential. So the basic ideas, randomization remains standard. Over time, not in the reflection paper, but it was due to the fact that I had to think about other stuff as well. I realized that maybe some of the innovations that we have high hopes for, decentralized trials, better endpoints for particular diseases, if you do a single arm trial, there might not be any history of that novel endpoint. So no context to refer to. If you think, well, maybe I can do partly also in single arm trials, assessment decentralized, yeah, you become dependent that it's entirely the same as doing the same assessment, not decentralized. If you lack a concurrent randomized control, that may actually also introduce bias. So consideration that came to my mind, actually be aware that possibly doing single arm trials might actually hinder some other innovations that we also would love to see happen. So but it's not in the reflection paper. It's a personal thing that I realized. Build up of the reflection paper. There are essentially two parts in the reflection paper. have a fairly precise definition of the concepts we need to describe, let's say, the main considerations. And there we have fairly strict definitions. And subsequently, the main part is to translating in that to more practical considerations in terms of single-arm trial design and assessment. So the first part, looking at first versions we rate, we kind of intermixed outcome and endpoint, etc. So we found the need to be precise on what we consider outcome, and that's simply what you assess at individual patient level, whether he or she survived or not, particular score, and then subsequently statistical summary measures can combine that into individual outcomes, like for instance the percentage patients which were cured or died or did achieve a certain lowering of symptomatic treatment results. We don't call them responders because that's a causal attribution. So it's really the percentage and not, we avoid anything in terms of responders because that's already attributing causality to a treatment. Estimates, equally important, relevant. Very brief statement is it's a little bit more difficult to apply without a control condition. Importantly, the treatment effect of interest, we follow essentially ICH E9. And there, both the attribution is there to actually treatment, but it's also there that the relative, so compared to control, is the effect of interest. So that also holds for a single-arm trial. In principle, the comparison of the summary measure to the population not treated, the counterfactual, and if you like, that's the treatment effect estimate of interest, also in case of the single-arm trial, and we are immediately aware, but it's a little bit difficult to derive only on the single trial as by itself. Probably the most, I think the most important definition or concept that we use in the reflection papers, isolation of treatment effect, and that has to do with causality. And we've been very strict in the definition to also be able to be clear on, if you go to actual practical conditions, where do the uncertainties start to come in. So the very strict definition here used for isolation of treatment effect is an observed individual outcome on an endpoint, survival after 10 years. As a bad example, but I will come back to that, can never occur without active treatment in any patient. So we've defined isolation of treatment effect at the individual patient level, and not because the average effect is much larger than you would expect in the general population or under current standard of care, but really the strict definition of isolation of treatment effect is really at the individual patient level. And this is very strong. It really requires knowledge of the clinical context. I think one of, you can imagine that SMA is a disease without treatment. We're quite aware that how an individual baby will actually develop and how short their lifespan will be. If you have a treatment which for that individual patient gives a totally different result, then you are close to isolating a treatment effect. But that requires quite specific knowledge of the clinical context and no doubt on causality. It's of course quite exceptional. Certainly in this extreme, it's really exceptional because you are, in real life, and that's where the uncertainties come again. There is some variations. Small miracles do happen, so some patients do have a better prognosis than others. If you start measuring, there will be measurement error. So there are lots of things that will actually add uncertainty to this causal attribution. Now, the treatment effect estimate, if you move in this direction and keep it as a concept, it's compared to, if you really isolate the treatment effect, it's actually comparing to no effect at all. And it's probably also immediately key that the estimate is hugely impacted by patient selection. So anything prognostic favorably will start increasingly look like a treatment effect. So, but this is a very, let's say, I would call it a theoretical strict definition, but that's to be aware on what are we actually losing in terms of increasing uncertainty if we deviate from this. A couple of other concepts. Internal validity we've defined it here as the treatment effect estimate from the single-arm trial versus the treatment effect estimate that would have resulted if we had a matching RCT, so a matching control trial. And external validity is the systematic difference from the treatment effect from the single-arm trial to the target population. The latter is, of course, also a problem in RCT. So they are not random samples from any population either. Also there we have a translation to the target population problem, but the point made here is that that's only due to if variables or prognostic variables are predictive. So that's generally more robust than you would think. But in a single-arm trial, everything prognostic will have the same effect. So the problem that's the statement there is really larger in single-arm trials. I will speed up a bit and skip on some smaller stuff. Translating into practice, we go through, it's in a couple of areas, so choice of endpoints, etc. The choice of endpoints, isolating the treatment effect is primary, but to just take an oncology example, in solid tumors, tumor response might indeed be able to isolate the treatment effect, because even in the response definition, there is also quite a bit of margin already included to deal with measurement error. But that might not be the outcome that's really most reflecting the clinical benefit. So endpoints are an issue, and isolation in a single-arm trial, isolating the treatment effect has priority, but we are well aware that it might not be enough. So you might have a challenge in an endpoint that's isolating treatment effect to be able to actually describe the clinical benefit. And this is therapeutic area, so that's the statement we make. And it's not exactly a balance between the two. Isolation is first. Otherwise, we simply don't know whether the treatment works or not. And only for working treatments, I'm interested in an estimate for the target population. Time to event, that's why it was a bad example. They are specifically difficult for single-arm trials. So the general idea would be that that will probably not fly. And I think statistically the issues are quite well known. It's time to survive. If you look at time to death, that's not only treatment. The disease course might be not so predictable, and the starting point, counting from which moment are you looking at survival, can be quite difficult and subject to bias fairly quickly. And then it's difficult to interpret. There are some exceptions. So diseases that start at birth may have birth as a proper time zero. So there are exceptions here, but in general, this is probably not going to help single-arm trials. We have similar problems with quantitative variables. Regression to the mean, measurement error may actually all contribute to bias, and you're not looking at the treatment effect, but you're looking at random fluctuation. Given this time left, so target and trial population, I think there we are quite concerned about the potential selection bias. So essentially the selection of patients that goes into the trial determines how plausible it is that this actually is reflective of what will happen in the target population. And I mentioned already the prognostic variables, and you can't distinguish that from prognostic from predictive. And the point rate, of course, it's always important to have an account of the screening and selection, but here it actually becomes crucial because the selection may actually determine the result of your trial. So it's emphasized that that process needs to be even more transparent than we are used to. Then the role of external information, also on purpose, not external evidence and not even external data. It can be usually general knowledge about the natural disease course. External clinical data can also, of course, also be used. It is critical for interpretation, but for the reasons I mentioned earlier, to actually understand what would happen under normal conditions from a clinical point of view. But if you consider to do this quite extensively, there's no one size fits all. And that relates to, you actually have quite a lot of very different designs on how you could use that. So that's seek scientific advice, and after this morning, seek scientific advice early and have an open discussion. Very quickly, the statistical principles, but there I would think we are pretty much aligned. Pre-specification, even more critical, because everything's open. And adherence to study protocol, no stuff happening in between that's not planned. That's much more critical than in an RCT. And usually in terms of analysis set, the full analysis set, but we have seen examples where that would actually bias in favor of clinical benefit, because subjects were included that were actually maybe not really diseased. And then you would probably rather exclude them. And a minor, a larger point, I will skip some, but we see examples with threshold, if you like, threshold crossings, defining a threshold. If you move in that direction, actually the uncertainty around that threshold should actually be part of the inference you make, because that's usually not a fixed value. And to end up, and I'm a little over the 5 minutes. Oh, 1 minute left. I'm pretty okay. So this summarizes what we've added is a more extensive table of sources of bias and potential remedies. So this is much more geared towards prospectively designing a single-arm trial. I will not go over them. It's a fairly long list. And most of them will actually be quite familiar to you. And the remedies, yeah, they might actually, they are not cures necessarily in the sense of remedies. They might reduce either the risk or the bias at all, but I think it's important to take them into account. And we are very, yeah, I think there are some comments already getting in. We've presented it more, but we are quite, yeah, let's say we are prepared for maybe not 1 page of comments, but quite extensive ones, because we know it's a fairly contentious area, but I think it's really important to engage in the discussion. So we're looking forward to the comments that we will be receiving very soon. And no doubt we will be back. Thank you.
Any questions for Kit before he tries to race off the stage? There will be opportunity maybe at the end, but if there are any 1 or 2 questions now. Question over there. Thank you for my runner.
So thanks for the nice presentation. So I have a very short question regarding the scope of the reflection paper. Are non-interventional studies, non-interventional post-authorization studies, are they also in scope?
They are not in scope.
They are not in scope.
No, we looked at studies before market authorization and designed as an interventional study, but single arm.
Okay, thank you.
Time for 1 more question at the back there, but higher up.
Yeah, thank you for this very interesting talk. I was wondering, in the context of single-arm trials, it's as you mentioned, it's very important to have external controls to provide context. I wanted to know your opinion on having, for example, in a single-arm trial, a non-randomized control arm to establish that evidence of.
We tend to be very precise in wording. I think I've pretty much avoided external control data. I've used external information and external data. And I think, and I want to go back to one of the remarks I made earlier. I think if you describe it like that, you're describing, in my view, a different design. And you would have to write that up prospectively in a protocol, also where the non-concurrent control will come from and which data that is. And then we are looking at a different design. if I'm in a medical ethics committee, I think the design with only the single arm there, or I have a protocol where all of this is also described, they are not the same design. So that design is out of scope.
Right, but then, for example, in that example, if you have an external control arm, an external comparator from historical data, for example, there are some timing bias, right? That it may standard of care changed, but if you incorporate kind of controls, concurrent controls in your design, you may mitigate some of those biases.
Yeah, but I think there are better designs than single arm trials.
Yeah.
Also, that are not immediately RCT. I really want to keep this discussion clean, because if you move there, the number of designs you can look at is probably infinite, which are very different in nature.
Mm-hmm. Yeah.
Thank you. We've got one question online, Kit.
Yeah.
You've got to go with that. Sorry. No. That.
Could you come back to the choice of outcomes? You mentioned that time to event and continuous outcomes are problematic. Would you say that only binary outcomes should be preferred for single arm trials?
I'm kind of trying to stay away from such sweeping statements, to be honest.
And yet,
we've kind of seen it all. Of course, we have seen examples where you could be convinced by a time to event endpoint. I mentioned if your time zero is clearly defined, it's a disease at birth. So it's not totally black and white, but it would not be my starting point, because so most of the things that I did see will probably be binary, and will be more of a disease or a cure status. So not binary because you've split a continuous outcome in two, because that inherits the same problems.
Thank you very much, Kit. We'll move on to the next presentation.
Thank you, yeah. So it's about time I introduce you to Pallavi Mishra Kalyani and Mark Levinson. They both work at the Office of Biostatistics in the Center for Drug Evaluation and Research in FDA. And today they're going to talk to us about regulatory issues with the use of external controls and the US FDA real-world evidence program. Mark, over to you, and you can share your slides.
Okay, so thank you. Let me spend a second sharing my slides. Okay, can you see my slides?
Not yet. We see your desktop, I guess.
You see what?
The Zoom cloud meetings. Maybe you're sharing another screen?
That's probably right. Let me try again. How's it look now?
That's great. Thank you.
Okay, I'm afraid I'm going to have to look to another monitor, so you may not see me looking at the camera. But let me get started. So first, I'm happy to be talking here today, and I thank the workshop organizers and the session organizers for inviting us. So I'm going to spend just about 5 minutes giving a quick tour of the FDA real-world evidence program, and the majority of our time, Dr. Mishra Kalyani will talk about relatively new external control guidance. So let me get started. Just to make sure the slide advanced. Good. I see a lot of you shaking your head. Okay, so real-world evidence in some ways is nothing particularly new at the US FDA. We've used it in safety for as long as I can remember in pharmacoepi studies and case series. But what changed is an emphasis, or at least a focus on using real-world evidence for efficacy. And that really got started in 2016 with this passage of the 21st Century Cures Act. This was a large piece of legislation in the US that did many things, but one of the things it did was called upon FDA to establish a program to evaluate the use of real-world evidence and to support a new indication for a drug already approved or to satisfy post-approval study requirements. The 21st Century Cures Act made it clear that there was no change in the standards for efficacy And our usual standard for substantial evidence for efficacy in the US remained unchanged. In 2018, FDA issued a framework for our RWE program. And although that's almost 5 years old, it's still very, I think, accurate description of our program that includes guidance, development, internal processes, external engagement, and demonstration programs. Our user fee acts also had some commitments related to RWE that in some ways overlap 21st century cures. And during this time, since 2016, we've issued a number of draft and final guidances, which I'll talk about shortly. And our latest user fee program, PDUFA 7, has a new initiative called the Advancing RWE Initiative, which I'll also talk about in my short time. Okay, this is a table or a figure from a paper by Dr. Kincado and Dr. Corrigan-Correa about the uses of real-world data to generate real-world evidence. And I think the most important point here is that FDA considers that real-world evidence can come from observational studies, and I think the rest of the world agrees with us there, as well as from randomized controlled trials. It just depends on the source of the data, not the design. And what this figure shows is sort of increasing reliance on real-world data. So even traditional randomized trials may use real-world data in assessing enrollment feasibility or trial feasibility, the selection of sites, but you may make more use of real-world data in a randomized setting. In a clinical setting, you might use real-world data to get outcome data or other kind of clinical data necessary for the trial. Moving towards the right of this figure for more reliance on real-world data, you may have a single-arm trial with an external control where the external control comes from real-world data. And finally, you have the fully non-interventional observational studies on the right. Okay, so this is a summary of our guidances to date and maybe a few upcoming in different categories. So we have 2 draft guidances on data considerations, one on electronic health records and claims data, and one on registry data. These guidances are sort of independent of the design, whether you're doing randomized or non-randomized or external control. These are kind of data consideration guidances. We have a guidance on data standards. In the US for submission data, it's CDISC, and there is not an exception for real-world data. So submitting real-world data to the FDA has to abide by CDISC standards, or you can read more about the details in this guidance. The next guidance, which I'll abbreviate as regulatory considerations, I think this is one of the more kind of interesting guidances because it's not so much like best practices in general for pharmacoepi. It's specific for regulatory considerations. I don't think you'll find this material anywhere else, and I am going to spend a little more time talking about that. The next set of guidances are design considerations. Dr. Mishra Kalani will talk about the external control guidance, which I think is a very interesting, extensive, and long sought-out guidance from FDA. So I'll try to be quick so you'll hear the interesting stuff. And then upcoming are 2 guidances, one on RCTs in clinical practice setting, kind of OWL phraseology for pragmatic trials, and one on non-interventional studies. And then there's our sort of a procedural guidance on submitting applications that have RWE in them. So there's a lot of guidances, and I think they're all very useful. Let me focus on this regulatory considerations guidance, because I think this is unique. This is not just kind of best practices for pharmacoepi. This is very specific to regulations and the US expectations. So this guidance is chiefly about non-interventional studies, although it says a word or two about interventional studies, and it discusses the need or the lack of for an investigative new drug application. It also encourages sponsors to engage with FDA early in the drug development process if you're using RWE. Naturally, sponsors should provide protocols and statistical analysis plans as they would do in a traditional study. But what is kind of a wrinkle here is to develop protocols and SAPs that use RWE. Some of that data already exists. So there's a some of that data may actually be kind of reviewed before you develop the protocols and SAPs, and that potentially can create problems. So FDA wants the sponsors to document all analyses performed on the data during the study, including the feasibility evaluations up front. And I think what's important is we have the same expectations about submitting patient-level data that we do for traditional RCTs. So if this is a supportive marketing application for efficacy, it's almost certainly going to require patient-level data. There are regulatory exceptions, but they're not unique to RWD. And the guidance also discusses sort of study monitoring, data traceability, and safety reporting for RWD. Okay, so my last content slide is on this new program, Advancing the RWE program. This fulfills a commitment under our latest user fee program, PDUFA 7. This is similar to other programs you may have heard of, like our Complex Innovative Design program. But twice a year, FDA solicits sponsors to submit applications to this program. If a sponsor is selected to the program, what they get is 4 additional meetings with FDA to help them develop the RWD study, the RWE study. What FDA gets in return is the ability to talk about the key design elements before the drug is approved, or if it's ever approved at all, or before the post-marketing study is complete. So that's kind of unique. You know, we're not supposed to talk, we're not allowed to talk about studies or information from non-approved drugs. But this is there's sort of a disclosure agreement that allows us to talk about it, so to create sort of more community learning from this exercises. There's only a few openings to this program each year, but all existing procedures for sponsors to engage with FDA continue to be available. So it's not like you can't contact FDA in a traditional manner to request meetings to get feedback on your program. So just for people interested in a kind of additional resources, the first link is for a general RWE site. It has a lot of information. The second link is for this Advancing Real-World Evidence program that I just spoke about. If you have questions, there's an email that's provided here. And with that, I'll say thank you, and I'll turn this over to Dr. Mishra Kalani to talk about external controls.
Thank you very much,
Sharing.
Dr. Levinson, and I'm going to go ahead and share my screen. Please let me know if you're able to see it. Maybe it didn't work.
Not, we can see it now, thank you.
Oh,
Can you hear us? No, there seems to be an issue.
I can still hear you. Pallavi, can you hear us?
We can hear you, Mark, but we cannot hear Pallavi.
hi, I'm sorry. Somehow my Zoom totally froze. Apologies. Hopefully
We can hear
that'll
you well now.
technical glitches. Can you hear me now?
Yes, all good.
Great. And you can see my screen as well?
Yes.
Wonderful. So thank you, Dr. Levinson, for the great preview to the RWE program and a discussion of all of the wonderful programs that are coming. I wanted to take the time today to briefly describe a new guidance that came out this year titled Considerations for the Design and Conduct of Externally Controlled Trials for Drug and Biological Products, which is a draft guidance for industry. As I previously mentioned, my name is Pallavi Mishra-Kalyani. I'm in the Division of Biometrics 5 in CEDAR, which is the group of statistical reviewers for solid tumor oncology products. So I don't
I think, yeah, maybe Pallav, if you can hear us, maybe if you try to perhaps turn off your video, I don't know what else to recommend. I guess Pallavi cannot hear us now. Or you can use Mark's Zoom.
I'm so sorry, I'm not sure what's happening on my end with the Zoom, but it keeps crashing. I wonder if I can join from my phone, if someone else can present the slides, that might be a better way to ensure that it doesn't keep crashing for you all.
Okay, we can share the slides.
Okay, I'm going to rejoin this meeting from my phone. I may not be on video for that reason, so my apologies, but at least you'll be able to hear my voice.
That's okay.
Okay, give me one more.
Well, these things happen. Yeah.
Okay, I'm on.
We can hear you and we'll try to share your slides now.
Yes. I can see the slides on the screen. Perfect. And then I'll just indicate next slide. Apologies again for the technical difficulties. I'm not sure why my Zoom keeps crashing, but hopefully this will allow me to at least complete the presentation. Next slide, please. So as described, again, Mark, I think described many of these types of guidances previously, but I just wanted to touch upon them again because I think we do have many guidances. And certainly while they are applicable specifically to FDA submissions, I think that there's also a lot of just very helpful and relevant information in these guidances regarding studies that include real-world data, selection of data sources for these types of observational or non-interventional studies, as well as study design and conduct guidance. So certainly they're very helpful for any FDA or US regulatory submission, but I also think that there's just a lot of great information that has come from multidisciplinary teams who've really thought about these issues and how best to conduct studies. Next slide, please. So as described earlier, I'll focus mostly today on the external controls guidance. I don't want to go line by line through each of the sections of the guidance, as you know, I think that there's a lot of very important information in the guidance itself, but I'd like to give just a good overview of what to expect in each of the sections of the guidance, so that if you're interested in learning more about a particular area, you can read either the whole guidance or just that section for a better understanding of the types of considerations we've provided. So this is the table of contents of the guidance. And if we go to the next slide, please. We provide information in the first two sections of the guidance, particularly about just an introduction to what is considered an externally controlled trial. And the scope of the guidance, which I think is critical because certainly, I think the question came up, in Dr. Rose's presentation regarding inclusion of external controls versus external data for regulatory decision making. And the concept of using external controls is quite broad. It can be external control data can be used in many, many ways. And because there are so many different types of trial designs, we had to really focus on very specific types of designs to discuss in the guidance. And in particular, we're really focused on external control designs that are intended to establish efficacy of a drug. So basically, what types of data may comprise external controls, the suitability of such trials, depending on the clinical context. So that may include the disease area and the type of treatment that's being considered. We don't really discuss external controls as summary level estimates. Really, we focus on the use of patient level data. I think this was also reiterated by Dr. Levinson in his discussions. And then finally, we don't discuss the reliability and relevance of the various sources of real world data. And I think that's critical because that's really discussed in the other suite of guidances provided by FDA and is very important in the consideration of a specific data source for an external control. But because that's such a broad and important topic, we don't repeat the discussion from other guidances and rather redirect the audience to read those guidance as well. Just briefly, I want to mention that the last section provides considerations to support regulatory review. In particular, the FDA requires access to patient level data. So it's important that that data may be submitted to FDA for our own analyses and review process. And also the data provenance must be available such that audits of the data are possible for the inspection process. All of this is described in section 4 along with descriptions of the discussion process with sponsors to develop any externally controlled protocol, in that we really encourage meetings with the FDA early and often. I focus on the first and last sections first because what I really want to describe is on the next slide. So next slide, please. What I would consider the meat of the guidance, which is section 3. Section 3 discusses the key consideration of the design and analysis of externally controlled trials. There are 3 major subsections. I could probably spend an entire presentation on each of these subsections, so it's incredibly difficult for me to boil these down to key points, but I've tried to do so here. Again, I highly recommend that if you have the time that you review the guidance document, because I think it is very helpful. But the 3 main sections, subsections include design considerations, data selection, and analysis plan. So in brief, the design considerations discuss the development of a protocol, which should be finalized before initiating the externally controlled trial. I think what this means is that in order to be best blinded to any outcome data, it's good to have the protocol ready and available and finalized to the extent it can be prior to the initiation of any experimental arm. The control data may already be available and you may have some knowledge of characteristics of that control data. And in fact, you must have some knowledge of characteristics of the control data. Otherwise, you will not be sure whether that data are fit for purpose or not. But if the outcomes are known for both data sources, it's much easier for bias to creep in the design of the protocol. And so it's best if at least the experimental data
is not available. The outcome data for the experimental arm is not available when finalizing the external control trial protocol. The SMN framework is really a wonderful way to design, a wonderful approach in designing the protocol and analysis plan. Certainly, this is a framework that's used across the board for many different types of trials, but it's no stranger to those of us from causal inferences. I think, you know, the SMN framework is something we really have to deeply consider in many of our observational studies. And it should be considered here for externally controlled trials as well. It's important to pre-specify plans for measuring and analyzing confounding factors and sources of bias. So that includes descriptions of how the analysis populations will be compared for similarity to ensure that they are sufficiently comparable to enable an analysis of treatment effect across arms. I think, you know, having actual metrics or actual measurements of similarity will be critical for regulatory understanding of similarity. Certainly addressing pre-specifying biases that may be of concern, such as, you know, immortal time bias or other expected sources of confounding to your best ability are critical in the analysis plan or in the protocol, how they will be addressed and then how they will be analyzed in sensitivity analyses is very important. Certainly there will be issues that may come up in the conduct of the externally controlled trial, and in the analysis plan there may be things that are uncovered, and certainly those should also be addressed. But to the best extent that we can pre-specify methods to avoid bias, that's the best way to ensure a well-developed externally controlled trial. Data selection for the external control arm is critical. As we mentioned earlier, finding data that's fit for purpose and has those qualities of reliability and relevance is very important. And on the next slide, which we'll go to in just a moment, we have a really wonderful table in the guidance that is pretty comprehensive in describing comparability considerations. I should note when we get to that table that, you know, it's not all encompassing. Certainly there may be other factors that should be considered on a case-by-case basis, depending on the clinical context. But this guidance, I think, does a pretty good job of describing the major sources of potential bias and comparability considerations across the populations. And lastly, I should note that our analysis plan section is not overly specific, and this is intentional. You know, the FDA does not really endorse any particular statistical method for externally controlled trials, and that's intentional. There are a lot of different approaches that may be sound and may be appropriate, and it's up to the sponsor to identify the appropriate method for their particular situation and then to justify it. Trying or specifying alternative primary methods is also really a good idea that should be included in the statistical analysis plan to show the robustness of the results, along with thorough sensitivity analysis plans, plans for missing data. And there should be, as I mentioned earlier, a formal evaluation of comparability. So next slide, please. So I apologize, the text on this slide may be slightly small. I hope you can see it, but certainly you can also refer to the guidance for, you know, easier reading. But this is the table of data comparability that I referenced earlier. As I mentioned, there are many different areas to consider in terms of making sure that the external control data are similar to an experimental arm, including time periods, geographic region, diagnosis, prognosis, attributes of the treatment of interest, including things like concomitant therapies, sorry, that's under other treatment-related factors, but all of these things together are very important to qualify so that when we do find a difference across populations, we can attribute it to the intervention of interest. Follow-up periods, intercurrent events, outcomes, and missing data are all critical considerations for data comparability. And if you are able to spend some time reading this table, I think it really provides a very thorough picture of what to consider and how to consider when identifying appropriately comparable external control data source. Next slide, please. So I just wanted to acknowledge the various groups that were a part of this guidance creation. This was led by the Office of Medical Policy in FDA CEDAR, but were many people from other parts of the agency who contributed to this. Certainly those of us from the Office of Biostatistics in CEDAR, but also individuals from the Office of New Drugs, Regulatory Policy, Surveillance and Epidemiology, and along with some people from CBER and CDRH and OCE, which are different groups at the FDA, not under CEDAR. We had this guidance publicly available until for comment until May 2nd of this year. So that public comment period has ended, and we're in the process of reviewing the comments that were submitted, and we'll consider them in finalization of this guidance. So it will be reissued sometime in the future as a final guidance for industry. That's it, and thank you very much for your time today. I'm happy to answer any questions.
Thank you, Bella. In the interest of time and to provide also the opportunity to our other speakers to present, I think we can skip questions for now for our FDA colleagues, and we can save them for later on if that's okay.
Okay, thank you. And so then we move on to the third presentation by Emmanuel Zuber from Novartis. He's a stats leader working in global drug development with loads of experience working primarily in oncology, hematology indications. So welcome to Emmanuel, who's now going to tell us a little bit more about some experience in real life of trying external controls for approval and labeling. 2 case studies. Over to you, Emmanuel.
Thank you. Thank you very much for the opportunity to present and for the introduction. So slides will be coming up soon. No worry. So this presentation is actually meant to tell you 2 stories, 2 real stories, illustrating the adventurous journey of using external controls for approval and labeling, and trying to figure out when it is worth it. I want to acknowledge the work of the teams who did this, extremely intense work. And also I want to acknowledge them for great ideas, suggestions, and a lot of material for me to digest, also to present. And I also want to acknowledge Rob Hemmings for great feedback and insights in preparing this presentation. So approval and labeling can be considered as a newer playground for the use of external controls, acknowledging that it's not new in other areas of regulatory decision-making, orphan designation, trial design and planning, of course, and pediatric plans and so forth. But here, the focus really is on approval and labeling. And those 2 examples are meant to try to identify guiding principles from those real interactions, and also discuss remaining uncertainties that would be good to get clarified.
I want to say a little disclaimer here. First, this presentation is based on real Novartis stories. Why Novartis? Because I'm there and it's easier to have access to the story, the full story. And then they are based on selected quotes from the regulatory interactions. So I hope those selected quotes are not too biased in the selection and that they reflect properly the feedback intents that was given. And last, I want to say that this is not to revisit cold cases. It's not to challenge or question the decisions that were made at all. It's really to try to derive something more general and to raise questions that I would love to hear our regulatory colleagues on. So I will present one case after the other with background, the external controls and the regulatory outcomes, and then get to some questions and conclusions. So let's start with Kymriah. And by the way, it's a bit busy, but with the yellow highlights, hopefully you can follow the gist of what I try to say. So Kymriah is a cell therapy, and that was already approved in 2 indications before considering the one subject of this talk today, the follicular lymphoma indication. And in follicular lymphoma, last line or late line, we consider that as an incurable rare disease, but there is a diversity of treatments available with limited efficacy, limited complete response rates around 15%. So the scientific question of interest here was, can Kymriah demonstrate a complete response rate significantly greater than, say, a null hypothesis of 15% in those patients? Of course, quite important other question of interest would be, can Kymriah improve overall survival over standard of care in those patients? And as we just heard a bit earlier, that might not be addressed the same way, those 2 questions. But the plan that was proposed here was to go with a single-arm trial called ELARA, and to see whether that could work to get this indication registered. So 2 scientific advices unable to understand that given the unmet medical need, the novel mechanism of action, and the promising data, the early data that was presented at the time, overall, an uncontrolled trial may be a possible basis for authorization, with the expectation clarified that the effects should be exceptionally compelling, right? On the FDA side, we had a pre-submission meeting and similar feedback. The data would be sufficient to support submission in the context of an accelerated approval. This is related more to the type of endpoints. Response rate and durability of response would be considered adequate in that setting. So that was supportive to move forward. But now we had, when consulting with the EMA reporter at the time of the protocol review, we had an interesting unprompted feedback. When reviewing a single-arm trial design, the feedback referred to the use of an external control design. So that refers a little bit to what Kit was referring to before. And clarifying that in such designs, the effect is expected to be dramatic, and therefore the null hypothesis should reflect how dramatic the treatment effect should be. So the question here was on a null hypothesis, but referring to an external control design. Now, it was not by a mistake of reading the protocol, as clarified later. It was further specified that being a single-arm trial, we assume that the external control will be pre-specified, including, by the way, having access to individual patient-level data for those external sources. And then that's the, acknowledging as well that the external control is not specified as of now, but is expected that it will be specified in a substantial amendment. So the way the sponsor took this is that, it's a bit unexpected to refer to an external control that was not part of the protocol, but we take it as a request to have an external control. And that's how we engaged to prepare for the EMA submission, preparing for 2 external controls and adding a systematic literature review on top of that. On the FDA side, what was the question that was raised is whether external control would add value, for example, to support regular traditional approval, in addition, of course, to the accelerated approval that could be supported by the single-arm trial. So the answer was no, and mostly, again, related to the endpoints, because the endpoint in real-world data would not be suitable in the context of a traditional approval. In that case, a randomized control study would be needed and would be deemed feasible, by the way, in this setting. So that was a very useful feedback at an early time, and that's why we didn't plan to include external controls in the FDA submission, and spared quite some work there. So as a first little wrap up here, we learned that complete response with duration of response could be enough to isolate the treatment effect with the demand that this effect should be exceptionally compelling. And a single-arm trial appeared acceptable for approval in both agencies. So that raises a subsequent question. So which part specifically will the external control play in the approvability decision beyond just confirming the relevance of the null hypothesis, right? It was raised in the context of changing the null hypothesis, but then deemed useful. Useful to what? And of course, the underlying question is, could the systematic literature review be sufficient for this? It's not the same amount of work, right? So we can discuss that later, hopefully. So the team went on and identified two main sources of data. One was a retrospective chart review in Europe and North America. Another one was based on electronic health records from Flatiron in the US. And there, quoting again the scientific advice, it was reiterated that contextualization with those indirect comparisons is considered useful to understand the efficacy of Kymriah in the current therapeutic landscape. However, it's only supportive, and then it's compelling results from the pivotal study, which will be required. There again, the inferential benefit risk estimation will be based on the absolute outcome. So again, that question, what will this be useful for really, right? As it is stated. Then the team went on to address all the different real-world data challenges that have been also presented in the previous talks. And I won't go into the details of that, but I want to say that it resulted in a 50-page section of the briefing book to describe all the aspects of how data sources were selected, how real-world patients were selected, what were the differences in patients, including exclusion criteria and endpoints. And then the team, actually, it's one of the first usage of the target trial framework combined with the estimate framework to really emulate a target randomized trial and identify all the areas of uncertainty that had to be addressed with specific sensitivity analyses that were also planned in the analysis plan. So I acknowledge this was not abiding by the recommendation this morning of brevity, but it was certainly complying with the recommendation of being specific and trying to really be rigorous in the way to address this external control approach. I won't go into, even though it's extremely interesting, the selection of index line. I invite you to go to the publication that is referred to here and the presentation, which is online as well, to get further details. But to give you an idea with this table, right, we had every component listed of the target randomized control trial that would be emulated with the two external data sources. And we had our strategy to address issues when there were uncertainties or disconnects, with highlighted here a few of the sensitivity analyses that were proposed. I won't go into the detail of that, but just to tell you, this was thorough. Results were pretty compelling in the opinion of the team, with a difference in complete response rates between external control and the study of 39% after the propensity score weighing here. And overall survival was also estimated with a hazard ratio of 0.20, again after weighting. So pretty good results, which led to a positive CHMP opinion, which interestingly, again, reemphasized that the contextualization of the findings provided valuable context and was deemed supportive due to the clear differences in outcome they show, right? So the treatment effect estimates seem to have been relevant or supportive here. However, when the team proposed that this could be part of the label with a table describing the treatment effect estimates in the different endpoints and with a little description of where this was coming from, what was the process of the external control comparison, the response was, no, this information does not describe a feature of the product, and the SMPC should only contain data on the product which is relevant to the prescriber. So that leads to the question, by the way, FDA did approve based on the trial alone, as we had initially mentioned. So it leads again to the question, what is necessary to make external control results relevant to the prescriber, especially external control treatment-based treatment effect estimates, right? And would that be different for the complete response rates and for survival, considering also what Kit was mentioning in terms of estimation from a single-arm trial as well? So, and then again, the same question, how critical was the contextualization here in the approvability decision process? Moving on now to the next example. Eltrombopag is a drug that was already approved in several indications, including in later lines of the severe aplastic anemia indication we're discussing now. The idea was now to move to the first line in severe aplastic anemia, which is still a rare disease, 2 per million in the Western world, and a life-threatening one. Existing treatments there in first line, yes, but still with low efficacy, 10 to 15% complete response, and that had not moved for the last 20 years. So the scientific question of interest was, can Eltrombopag combined with the standard of care, those immunosuppressive therapies, IST, demonstrate a greater complete response rate compared to the standard of care in those patients? A single-arm trial would be the basis here. It was an investigator-initiated trial from an NIH center in the US, which was expert in the field of this rare disease. And it was a regimen finding study with 3 different regimens combining Eltrombopag to the standard of care, human HTG and cyclosporine A, which are those ISTs, right? And preliminary results of that study had been shown to a big hematology congress, ASH, and had been viewed as transformative at the time. And that's what led to the idea, maybe, you know, we could get this registered. That's the study, 3 different cohorts with different combination regimens. The third one, where Eltrombopag was started from the start and lasted for 6 months, was the one giving the best CR rate. Well, we knew that at the end, of course. And was considered as the regimen for to seek approval for. So coming to FDA with a Type B meeting, can this study support label expansion? The answer was no. The effect of Eltrombopag has not been isolated, and basically the study does not provide a comparison of the effects of adding Eltrombopag to the ISTs. And so in the discussion, it was agreed that the sponsor would develop a plan for historical control. And then it was nevertheless clarified that the endpoints were acceptable, but we had to provide the evidence that the addition of Eltrombopag did contribute to the effect, right? So the team planned for external controls for the FDA submission, and then confirming in a follow-up Type C meeting, yes, this data, the study and the historical control is adequate to support the filing of an sNDA. In Europe, there was first a rapporteur meeting in 2016, which appeared similarly supportive in the team's eyes, saying that the 6-month data that were shown, that was preliminary data, were very promising, and that Novartis should demonstrate the improvement of this treatment setting in comparison to the standard of care. Fair enough, right? Again, a high-quality comparison of matched historical control data will be important in the assessment of the results. So a similar request formulated differently, but similar request. And the co-rapporteur further elaborating that the data could support the variation because of the compelling results that would not have happened without a true treatment effect. That was based on the preliminary data that was shown, right? But again, confirming that the historical control data would be important and ensuring that it is well matched to the pivotal study. 5 minutes left. We had compelling results, indicative of treatment effects, but we had to demonstrate the improvement over control. So again, a single-arm trial with external control appeared possible to support the expansion of indication, and that's what was planned for the EMA submission. I'll go very quickly there. A very thorough selection of possible controls, ending up with only 4 studies that happened to come from the same center. It's a rare disease, it's an expert center. So basically that was actually perceived as an asset because the internal validity of the comparison, if you will, could be reinforced by having the same investigators and same site working on this. And the rest was very similar on the endpoint, the population, the control treatment as well. Different analysis methods, whether on summary, I mean, study level summaries or on patient level data, matching, weighing, everything. Results are quite interesting and compelling as well. 44% complete response rate against the 17%, and that was very stable with all the different analyses done. It was also stable when you would broaden the historical control with studies that were not matching on the population, not matching on the endpoint, but effects as depicted here were still big. So the regulatory interactions with EMA were the following. A first set of questions, 3 major objections and 22 other objections, raised the fact that while the search criteria seem reasonable, the efforts to match patients was acknowledged. Still, in the absence of randomization, it's not possible to exclude the influence of other known and unknown prognostic factors. Okay, I mean, it was clear from the start there would not be randomization, but that's the feedback received. In a second set of questions, 2 major objections left, 5 others. And one of the major objections was still that, again, the selection of the studies, the statistical approach can be considered acceptable. However, there are limitations, and those are, in the comparisons with historical control, referring to the bias derived from the lack of randomization. And clarifying also that in principle, one-arm studies could be considered acceptable if comparative trials are unfeasible. Also related a bit to one point you made earlier, Kit. Last round of questions, all the other objections had been resolved with a number of sensitivity analysis and so on, but one major objection left: the lack of a robust comparison against the established treatments. The indirect comparison with historical data cannot overcome this deficiency. So the key question here is was this based on the principle, right? And if the acceptability of external comparisons driven by the predefined, well, is that driven by predefined general principles? For example, was the initial question of interest to compare a combination treatment in first line with a single-arm trial and historical control, was that as a principle impossible, which could be the case, but then could that feedback be provided at the stage of early consultation? So that's really what was very puzzling. The team went on through a first oral explanation, similar feedback given. I'm trying to rush here. Absence of randomization. There were 2 divergent opinions, though, saying that the totality of the data could justify an approval and a randomized trial would be difficult to run. But again, that was a minority of the votes, of course, as you see here. The team requested a re-examination. Similar feedback received, went through a scientific advice group meeting, went through a second oral explanation. Ultimately, same feedback given, almost word for word, leading to a negative CHMP opinion, right? On the other hand, FDA did approve it on the trial data and historical control. So here the questions are, how could the appreciation by the sponsor of the likelihood of success of the use of a single-arm trial with external control be better guided before filing, right? And then is the consideration of the feasibility of a randomized study expected to remain as critical with evolving external comparison and data sources and method? That's a bit looking forward, but it's as this criterion also came up in the review process, right? In conclusion, so the use of external controls in the approval and labeling process is still new, and I think we are all learning, right? And that's the purpose of this presentation. I really want to emphasize the significant time and resource investment on both sides, right? Also referring to our discussion this morning, if a reviewer only had 1 day to answer the many questions and review the 50 pages at the agency, I have a lot of compassion for handling this, right? It's taking a big space in the regulatory discussion, so it's better be worth it, right? So early guidance is valuable to ensure the dossier is adequate with the right sensitivity analysis and so forth, and also that any principled showstopper is clarified up front. I will leave you with a few remaining questions and recapping a bit what was said before. What does contextualization of the single-arm trial results really mean, knowing that the single-arm trial results anyway are expected to be compelling anyway, right? So more specifically, when is it not really necessary for approval to bring all this set of historical comparisons? And also, when is it not sufficient at all for approval anyway, right? Regardless of the quality of the data and so forth. A second consideration is what is necessary for sufficiently low uncertainty of the treatment effect estimates? Is there any way to appreciate that regardless of all the sensitivity analysis you will do, the level of uncertainty will remain too big? So is there a way to guide sponsors on that? And third, when is this contextualization really useful? Also to the patients and prescribers, what influences whether an estimate on a particular variable is relevant to the prescriber, and whether this can be included as well in the label. Thank you very much for your attention, and looking forward for the discussion.
Thank you very much, Manuel. So I think it's probably important that we spend a few minutes now, as there's loads of questions that the regulators could answer, but luckily for them, there isn't time to answer all of them. Kit, may I start with you? You can choose any of the questions there to answer, but I'm being kind rather than asking you the hardest one.
Yeah, it's probably. Maybe put the questions up because I. Probably start at the bottom because I think you kind of started the discussion and the examples with to what extent, even if asked by the regulators or by us to provide external data to contextualize results, it doesn't make it into the label. I think your last question, if I is what's the reasoning behind it? Is this considered? Yeah. It gives you some. So the term we are using is contextualize, and that's actually indeed trying to understand the results of the single-arm trial in the broader context of the disease. That's not unique for single-arm trials. That's also relevant if you run randomized clinical trials, but it's relatively maybe more important or more influential in the decision-making. Whether or not that information makes it into the label is maybe not so much a question on what is relevant for a physician or a patient in general. I think I made the remark also earlier, but it's also what are we presenting there? And the way it would enter the label and also the proposals we've seen is that the external control data is contrasted what you found in the single-arm trial. So and there is not a lot of nuance there. So in the EPAR, we can completely explain what happened with the external data, how it was used in the assessment, but that's not what's happening in the label. There you see a table, you see estimates of response rates or survival times, and you see them comparatively, essentially, between what happened in the single-arm trial and what you derived from the external control data, but without a lot of context how to interpret that. And it looks pretty much like as if you have run a randomized clinical trial, but the interpretation is quite different. And also the role, external data, is a lot, however thoroughly it's done, there are selections there which are not part of the trial design. And they can be statistically well done, but it's really, really different compared to I've done an experiment where I've made the internal comparison and I can immediately interpret what I see there in terms of comparative effects. And for me, that's much more the reason why it doesn't make it into the label, because you can wonder what information does it actually convey? And can I really have a reliable interpretation for somebody who quickly reads it because they have to take the medicine or have to administer the medicine? Is this really informative? And it's not the only context that you can drive from. And over time, it's complicated. Because which external data, if I pick a different set, I get a different table, and it might be equally well done. So
I don't know how much you want to enter the discussion, but yeah, I mean, and we did, I acknowledge we did discuss that a little bit yesterday already, which was based on the same example. Now, at the same time, it's in a context anyway where your single-arm trial results are meant to be compelling, right? But still, the contextualization was useful. I hear that it needs to come with the right caveats or the right aids to interpretation. But first, it does happen, right? It does happen in some labels, and that's why the question came as well. What does, I mean, is it just the randomness of judgment here, or is there anything that can guide the sponsor to anticipate whether it will happen or not? I guess
that's. I'm looking also at the years in which these examples play. So I'm going to immediately admit that there was a reason why we wanted to look at single-arm trial and have a reflection paper there, because we saw it happening and we did realize we might not be overly consistent there. So I think there are now two parts of two parts in the answer. It's the single arm trial guidance will actually help making that a bit more consistent. At the same time, we're also looking at the SMPC and triggered. I also think I mentioned that it's triggered by the estimate discussion, but it also kind of makes us reflect what's actually the information that we want to convey there. So I think moving forward, I think we can provide a bit more consistency and guidance on what can be expected also in terms of external data. But I would keep my hopes fairly low given the argument I gave before. I don't think we see massive representation of external data in the label because it's not directly related to the product. A competing product, should they present exactly the same external data? Or should we monitor if somebody comes with a competing product?
Well, it's an estimate of treatment effect. You could argue.
Question differently. You're Novartis, and we have also Roche. You're in the same patient population, but you decided differently on the external data. And you did both did a thorough job described in 50 pages. And what are we supposed to do? Each of you have your own
Estimator.
Own estimator, which is not related to the design of your study. Just as a thought experiment.
Yeah.
And that's where it differs from actually having a controlled experiment. Because then it's clear. So that's the kind of thinking I think you need to be aware of before something actually enters the label.
Right. I mean, we can continue, I guess, after the.
Thank you.
Worried now a bit.
Thank you, Emmanuel. There are many more questions, but unfortunately not sufficient time. We now move on to our last presentation. So welcome to the floor, Stefan Lang, who's going to give a HTA view on the EMA draft reflection paper and the FDA guidance. So welcome, Stefan.
Thank you very much. Okay, thank you very much for the invitation. I'm really happy to be here, and I joined very interesting discussions at the former session and also now. And I'm very interested whether it works with the presentation. So when we talked about this, my part here, then I was pleased to have a look on 3 guidelines. So don't be surprised if I'm talking about also the actual guidance from the FDA with regard to accelerated approval in oncology. So great, thank you very much. But it's not in the internet. But that doesn't matter. Okay. So we have a saying in Germany: the last one bites the duck, bite the ducks. So that's my thought that I decided to present as the last one. So I will go quickly through my slides. So we have enough time and enough space for discussions. I think this should be important because I can imagine that some of my, thank you very much, of my points are not too surprising to you. So this is a picture which all of you probably know, the typical life cycle. And we are not tired to we are not become tired to explain and to note that HTA and regulatory approval are very different things. And I know that you all know this, but we often hear in discussions that, oh, this was accepted by the regulatories. Why don't you accept this? So, and this is a reason why I say this is not an automatic mechanism. We have different questions with regard to HTA. And for all of you, you know that we in Germany have not this 4th hurdle and not the cost effectiveness only for information. This is still a very comfortable situation in Germany. So this is a definition of HTA from the UNETA consortium, and I show this because it clearly says that the question in HTA is how well does a new technology works compared with existing technologies, or as we say, standard of care. And this is mandatory. We are not interested if there are therapeutic alternatives in placebo-controlled trials. This is not interesting for us. It may be sufficient for the regulatory decision, but it's not sufficient for HTA. And we have slightly different views, I think, on endpoints. We have a very strong, stark interest in what we call patient-reported outcomes, PORS, which relate to morbidity and health-related quality of life. Of course, surrogates are acceptable, but only if first data on the really interesting endpoints are not available, and if the surrogate is valid and validated. So this is, I think, a very nice phrase from the reflection paper. It is the responsibility of the applicant to adequately justify to regulators or assessors why a single-arm trial provides convincing evidence for the regulatory decision. And the same, and on the other way around, it's not the responsibility of the regulators or assessors. It's not the responsibility of the regulators and assessors to justify why is that as a single-arm trial or an externally historically controlled comparison cannot provide clear pivotal evidence. It's not our responsibility to say or to justify why we don't think this is convincing. Sometimes we hear this, but that's not our job. I show you this phrase because it's a similar point. First of all, this is from the guidance on considerations to support accelerated approval in oncology. It's what I want to highlight here is that the requirement of pre-specification, that's very important. And this is also mentioned in the EMA reflection paper, that's especially challenging in molecular or biomarker-defined populations. This is a special problem. And if we are honest, in oncology at the moment, we are talking nearly all times about biomarker-defined populations. And in this situation, it's especially in particular challenging to provide convincing evidence from single-arm or non-randomized trials. And again, this is very important. Again, HTA looks at the comparison with the available therapies or standard of care. And what is an available therapy or standard of care is made at the time of the assessment and not at the time of starting the study. So when I read this, I was happy. I was really enthusiastic because I found this idea so fantastic. But now I read that this idea of isolation treatment effect was still in action in 2016. But I read it the first time and I said, yes, that's it. That's it. And then I thought about, and then I read the whole paper and all the shortcomings and what are the requirements. And I thought, First of all, as you know, we are not looking only for one primary endpoint in HTA. We have different endpoints which are the same interesting for us. So this requirement is necessary for more than only one endpoint, and this might be difficult, of course. This might work for one endpoint, but not for another. And what can we do then? How to estimate treatment effects for endpoints for which isolation doesn't work? So, but this is our need. We need the estimates for other endpoints, for example, for aspects of quality of life or patient-reported outcomes or morbidity or adverse events whatsoever. And I have, after thinking about 2, 3, 4 nights, the feeling that for HTA, this approach, no matter how convincing it appears, is only valid in rare, very rare, exceptional cases. Our experience says that we in Germany, in our drug assessments in the last 13 years, 12 years, 13 years, there are a handful examples where we came to a positive opinion by means of a single-arm trials. But it's really rare, and we have made about 1,500 assessments in this time. So you can calculate the rate. It's really small. Here are the reasons. One shortcoming, the question is, how can you control for this without an external control? Of course, you said it's not the intention. The trial has to stand for its own. But I'm just a little bit wondering whether this is really a realistic situation, a realistic option. Okay, here again, pre-specifying plans are very important. And what I would like to mention here and to highlight, what we also sometimes, or more than sometimes hear is, it is not sufficient to use the data that is already there. If you use, for example, a registry for your external control, but rather the data that is required. So, and here we are talking about important confounding factors, and there is a very critical question: how to identify the important confounding factors. How should you do this? Asking clinicians, thinking about clinical context. So there's one idea about this from Puffolete and colleagues, which they published in 2022. But this is a very, as the authors say, a very resource-intensive undertaking to identify the important confounding factors in the way they are describing it in the paper. So this is a very critical point. And we heard about this in the presentation from the colleagues from the FDA, that the so-called real-world data, they probably, not only possibly, but probably lack the necessary details. And we know that we have in such real-world data a lot of problems, not only with details, but also with missing data. So, and these problems which are described here will probably even more the case for the identified important confounding factors. Again, it's not sufficient to use the data that is already there, but the data that is required. So, but nevertheless, we were very keen to read these guidances or a reflection paper and the FDA draft guidance, because I think it's still in line with the European HTA view on these topics. And this is in the previous session mentioned guidance on direct and indirect comparisons, which is really advanced, I think, and really helpful. And here we are discussing the situation of the so-called simulated treatment comparisons or matching adjusted indirect comparisons. And these are the situations where sponsors try to compare their single-arm trial data with summary measure data from other trials. And this is nearly always not successful. And I'm highlight here that at least for those external control trials or data or non-randomized trials, we strongly recommend that full IPD data are used for these undertakings. So I give you some experience from the German decision-making body. Here I made this for another different presentation former this year, but it's rather actual. There were 21 procedures, 6 were cancelled after re-evaluation, and we have 15 decisions. And I show you what are the outcomes of providing the GBA with single-arm trials. So we are talking about orphan drugs, and in 12 cases, there was the judgment added benefit not quantifiable. Here you have to know this is a worst rating, because the situation in Germany is that orphan drugs, unless they reach a certain sales volume, are having an added benefit by law. So this is a privilege they have, and so you cannot say, oh, we see no added benefit. It's not allowed. It's given by law. So and if the GBA concludes we cannot quantify it because the scientific data do not allow quantification, this is the worst possible rating. So you see in 12 or 15 decisions that came to this worst conclusion. And these were all single-arm trials, and the reason was the populations were not comparable. It's very easy. And there was one situation, one decision, one procedure where they gave a considerable added benefit. This was based on an RCT, and second, where we had a major added benefit. And this was also a single-arm trial, but there was a very clever comparison with siblings. This is a special situation in gene diseases, but this was really convincing. And in the 15th case here, this drug underwent a regular assessment because it exceeded the threshold when they have to provide convincing evidence to IQWiG and GBA. So I give you one example to show the problems. This was a dossier for onasemnogen abibavobek. I think it's known under Zolgensma, the 2 million injection for spinal muscular atrophy. And here you see we have the requirement. It has to be compared to the standard of care. Thank you. This was to this time nusinersen or Spinraza. And they compared their single-arm trials for Zolgensma with here summary measures for nusinersen from publications. And it's really a very strong point estimate for the overall survival, but the comparison were unfair. Because for these patients with type 2 or type 1, type 2 SMA, it's well known that you have to treat the patients very, very early as possible. And in the nusinersen arms, there were a lot of patients who had a disease duration at the beginning of treatment, which was longer than 12 weeks, more than 12 weeks. And in the Zolgensma arm, all children were treated within 12 weeks. So, and if you restrict the analysis to children with treatment start within 12 weeks after diagnosis, then you see that the effect estimate diminishes, and there were additional problems because there were different in- and exclusion criteria in these trials. So at the end, we came to the conclusion that this does not provide sufficient or convincing evidence for an added benefit. So again, a phrase from the FDA guidance. So short note on the target trial emulation. It's also a compelling idea, but also difficult to realize, design a non-randomized study like a randomized one, but just omit randomization. It sounds very easy, but it's very difficult. As you know, you have to choose an alternative to randomization with consideration of the relevant confounders. You have to make sure that the observations start at comparable times, called so-called time zero, to guarantee the ITT principle. And you have to address impossibility of blinding. It's not an easy going. Again, pre-specifying, make a protocol about this, and statistical analysis plan, all this stuff also for external control trials or non-randomized trials. So conclusions. As I said, using non-randomized trial data as convincing evidence from comparative effectiveness is expensive and resource intensive, I would say, sometimes in a resource wasting undertaking. In many situations, this is a very nice phrase. In many situations, the likelihood of credibly demonstrating the effectiveness of a drug of interest with an external control is low, and sponsors should choose a more suitable design, regardless of the prevalence of the disease. The omission of randomization makes a rare disease not more prevalent. So this argument, I cannot run a randomized controlled trial because we have a very rare disease, is nonsense. I can randomize 10 patients, of course. That is a problem of power, but the power doesn't increase if I omit randomization. So, very important point, data quality is key and must have the same level as for randomized pivotal trials. Standalone single-arm trials may be convincing evidence for HTA, as I said, in only very rare, extremely very rare, extremely very rare exceptional cases. And last but not least, make use of IPD for externally controlled trials to apply adequate methods for confounder adjustment. Thank you very much for your attention.
Thank you very much, Stefan. Any questions to Stefan? Okay, let's go online.
This is very specific. What is the reasoning behind the strict separation in mortality, morbidity, and PRO, apart from the legal framework?
Oh, what's the reasoning behind this? Oh, I can make it easy because it's a formulation in our law. We have considered these endpoints, so that's it. It's only legal, but it's also useful. It's also reasonable.
Oh, thank you. We have a question in the middle there. Oh, one row out. It's close. There you go. Thank you.
Hello, thank you for the presentation. My name is Pei from Novartis. Just a question on the hypothetical scenario. If I want to develop a drug that is potentially used to treat the patient who suffer from nuclear power plant explosion.
From what? Sorry, from what?
Suffer from nuclear power plant explosion.
Okay.
Would you still request the randomized control trials? And would you accept a single arm trial? Thank you.
Yeah, okay, I understand. Maybe you know that there's a randomized trial for the benefit of parachutes. You know this? You know this? No, it's not a randomized trial, parachute against nothing, but of different parachutes, because we know that parachutes are working. But if we have some modification of the parachute, of course, I need a randomized control trial. So I know all these examples that there are situations where sometimes jokes that there's no need for an RCT. But as you know, in medicine, and I make this job, let me say, 40 years, this is a rare, extremely rare exception that you really, that you cannot run a randomized control trial because of ethical reasons, because you know that your treatment works. From my point of view, this is an extremely rare exception. So maybe I didn't understand your question correctly, but this is where would be my answer.
So if we have a randomized study, still there's a lot of kind of push towards having as low number of patients on the comparator as possible, very often. And so we don't go for 1-to-1 randomization. Go for 1-to-2, 1-to-3, 1-to-5, 1-to-10. How big can the ratio be, and what are your guiding principles behind that?
I have no idea, but I think it's not the topic of this session today. So I think the ethical issue doesn't become better if you change your ratio. It doesn't make a study more ethical if you choose not the 1-to-1, but the 1-to-10 ratio, for example. It doesn't make sense. So this is a different question. The ratio for randomization is given by your power, your thoughts about the power of the efficiency of your trial, not by ethical questions.
Just a comment first on Alexander's comment. When you do this 1-to-1, 1-to-2, 1-to-3, 1-to-5, 1-to-50, etc., your power goes to 0. When you go to 1 to infinity, maybe that's the single arm trial at the end of the day. Just to be a little bit careful with that argument. But Stefan, I think there are situations where these external controls are really meaningful and where you don't have an alternative. One example is when you are in chronic diseases or very slow progressing diseases, the value of a treatment is sometimes only really there after many years of therapy. And you can't, even when you do the controlled clinical trials, you cannot keep up with a randomization for that long. And then afterwards, for long-term follow-up efficacy analyses, I don't think you have an alternative. And then external control is better than just referring to literature, just as an example.
No, not to be misunderstood. I didn't want to say that always external control trials are not useful. They may be very useful, but I think the efforts are much more for a proper non-randomized trial or an external control trial in comparison to an RCT. People think mostly the other way around, but this is wrong. The efforts are huge. And this should one have in mind, should kept in mind. But nevertheless, I agree that there may be good situations to run a non-randomized or external control trial.
Thank you. There's a question over here.
Yeah, sticking a little bit with what Alexander started. I mean, looking at situations where you still do randomization, but you have fewer on the comparator, and then you do a hybrid kind of situation where you're augmenting what you're doing with external controls, so that you also have, you could say, the opportunity to, at least to some extent, assess the validity of the non-randomized part of the comparison. How do you view studies that use such a design from an HTA perspective?
Sorry, I didn't understand your question.
No, if you run, you could say, if you have, I mean, you randomize most patients to the active comparator, and then you have, when you're supplementing up with external controls, so you end up in this hybrid situation, so these augmented kind of studies. how would you view that? Because there at least you have some opportunity also of validating, you could say, the external comparison.
Yeah, I know this. This runs under different names. So you want to extend your limited knowledge from the RCT with broader data from non-RCTs or external control trials. Yes, some call this borrowing, or this is, but it's not new. But there's 30 years ago there was a suggestion that they called it. I just forgot it. The comprehensive cohort design. So this was an example in breast cancer for the question whether to for mastectomy against lumpectomy, and the people assumed that it would be very difficult to randomize a sufficient number of patients for this trial because of preferences, and then they undertook a randomized trial, but they complemented it with a cohort study. And of course, I think this can be very useful, but the important issue is that you have to come to the same or nearly the same results. So you get a problem there, they're different. The effect estimates are different, and you get a problem. But if you can show that's homogeneous, it's fine, yes. From my point of view, I had to say this is I have no such slide that I have a disclaimer that I can only talk for IQWiC, or so I think that's obvious. Yeah? I cannot talk for the HTA community. Sometimes I cannot talk for IQWiC because we have discussions within the agency, and not all my colleagues are the same as me. So, but it could be that some in IQWiC would say no, forget it. My point is yes, could be helpful.
Okay, may I people ask all our speakers to come in front? And there is a question on the chat that perhaps it's worth, going through. And I hope, Pallavi, you're still with us.
Thank you so much. And I'm going to combine it with the second question that's come in. So they're trying to understand the concept of raising the bar on estimates or on the endpoints for single-arm trials versus randomized trials. And similarly, when you get into a scenario like gene therapy, whether an open label randomized trial is superior to a single arm trial.
For whom is the question?
I think multiple perspectives would be great.
Okay.
Who wants to go first?
Happy to go first, and also happy to be brief. Also in the I think, yeah, single-arm trial is open label per definition. If it's formulated this generally, I think the answer is simple. Go for a randomized design. Because you already have, the single-arm trial is included in a randomized design. So essentially you have both.
I think I would agree.
And then with respect to the endpoint choice in a single-arm trial and the bar being higher versus a randomized controlled trial.
Oh, I'm not necessarily, I would probably not entirely agree with that formulation, that the bar is higher. I think the design forces you to have criteria there that you may not need in a randomized trial. But that doesn't, for me, that's not the same as the bar is higher. I think also endpoints are crucially important in a randomized design. But the criteria can be slightly different because the single-arm trial design brings particular problems that you need to solve. So I would not phrase it as raising the bar. I think it's an attempt, maybe phrase it differently. Being very precise in your endpoint choices is an attempt to at least move a little bit into closer to the bar that the RCT would pose in terms of level of evidence.
Okay, I think it belongs to the concept. So it doesn't make sense to speak about different bars, because without this definition, it doesn't work. So,
I mean, the bar has been made higher in terms of the extent of the treatment effects more than on the choice of the endpoints. which is a bit the ambiguity between requiring an endpoints that enable isolation of treatment effect as very strictly defined, but still requiring the treatment effect to be very impressive. So it's, in that sense, it's a double ask, right? Yeah.
Okay, thank you. Oh, Emmanuel, one more thing.
Actually, I had a question to Stefan. In the context where different HTA bodies, I mean, as you mentioned, the questions are often different than the questions from approval. And then there is the expectation for randomized ways to address those questions. And so if we step back and take a systemic perspective on the system and what is needed for innovation to reach patients, if you push it to the limits, you would need a randomized trial for every single HTA body to get the treatment made accessible. So could you comment on this?
If I understand you correctly, you say if we take the series, we have to run for France, for Germany, for Bulgaria, for Netherlands, for Belgium, each different RCTs. I don't know where from which this idea comes from, this idea, because I don't say that we have always the necessity to have for all these different needs, different RCTs. So you must think about maybe new kind of RCTs, maybe more than only 2 arms, maybe 3 arms with a common comparator. Maybe we should try to design RCTs that allow to make indirect comparisons. So this could be an opportunity. I understand, but of course we cannot expect that we have an extreme 27 different RCTs and for the European HTA.
This is 27 arms in an RCT, likewise, right?
Yeah, no, this is very extreme, and I know this discussion, of course, as you know, and we have to find a way how to deal with this, but I think there are possibilities, and you are the experts to show us convincing analyses with the convincing evidence that works. So it's up to you.
If we guide it, I think it should work. But
thank you very much on that note of a challenge to everyone. I hope you've enjoyed the session. Thanks again to all the speakers, including our online speakers. Now I'll hand you back to Hele to close the meeting.
So maybe I can start. Now this 8th EFSPI Regulatory Statistics Workshop has come to an end. And I hope you have enjoyed the workshop, the interesting presentations and discussions, and not least the opportunity also to network. I would really like to thank all of the speakers, the panelists, the chairs, the poster and short topics contributors. Thanks a lot for engaging in the workshop. And also a big thank you to the audience for taking part of the great discussions. I would like to emphasize, like Kasper did yesterday, that we highly appreciate this informal, friendly, engaging, and active discussions. And we really appreciate your willingness to share your views in an open and honest manner. So thanks a lot for that. So if you missed any of the sessions, don't worry. We'll do what we can to make slides and recordings available on the website. So please go in and check to find out when they will be available. I would also like to say a huge thank you to the scientific committee members. They have been working for a long time, very hard, to ensure that we could have such an interesting and engaging and relevant scientific program. So I hope you will all help me in thanking the scientific committee members. And not least, a huge thank you to the local organizing committee, Egbert, Uli, Christoph, Kasper, and Emmanuel. Without you, we would not have this workshop. So thanks a lot for that. And Uli, I guess there are two more persons we should not forget to thank, right? Four. Oh, actually four, okay.
Or they are the two admins with Bibi and Laurence. They stand outside. They didn't want to come in. But without them, it would be quite challenging to have such a meeting. And then I would also like to thank the Biozentrum. We had last week the CEN, this week the EFSPI regulatory workshop. There were two technicians coming in every morning at 7, staying till the last one left the building, etc. That's Gary and David, and even when they are not there, a big thank you to them as well. And before I hand back to Helle, I need to, yeah, Manuel is laughing, he knows what comes. The wine tasting, you know, we made a little game on who is winning, the German or the French wine. We had, as statisticians do very well, we had a metric how to win. That was the number of empty bottles. Interestingly, we ended with 23 empty bottles for France and 23 empty bottles for Germany. And when we just saw that, then there was an isolated bottle still standing around, which came from France, half empty. But that was not in the metric. And as statisticians, we started then a discussion on, did we have actually the right endpoint or not? So I would like to stop the discussion here. I think you got enough with the use of secondary endpoints to decide again. But I hope you had also a lot of fun today, yesterday with the wine tasting, and see you next time.
I would like to remind you to please fill in the feedback form. After the workshop, you'll receive a link to the feedback form, and it's really important for us to get your feedback so we can improve every year. So we hope you'll take the time to do that. And then I hope you all...
One last thing I nearly forgot. Sorry for that. But a big thank you to Helle for leading us through this successful workshop. Helle had probably most of the work getting the scientific committee, getting the local committee organized and on track. That was not always easy, but as you can see from the result, very worth and valuable. So thanks.
It was a great honor, and thanks for the opportunity to be involved in this. So I hope you've all seen that we have fixed the dates for the workshop next year. Otherwise, it's up here. It's in the same location, the 11th to 12th of September. So please mark your calendars. And again, I would like to remind you about our US counterpart, the Regulatory Industry Statistics workshop that actually takes place in just a few days in Rockville. And they also have announced the date for the workshop next year. So please keep that in mind. And with that, I would like to close the 8th EFSPI Regulatory Statistics Workshop. I hope you have enjoyed it, and I hope to see you again, all of you again next year. So I wish you a safe journey back home.